Hello everyone, my name is Juan Henao and welcome to my poster presentation about identifying dysfunctional mechanisms of pancreas-resident T cells in islet autoimmunity through single-cell immune profiling. But what is type 1 diabetes? This is an autoimmune disease characterized by the progressive loss of the pancreatic beta cells, the entities in charge of the insulin production. The main responsibles of this loss are the T cells. Therefore, we want to share with you our results about identification and characterization of type 1 diabetes related lymphocyte clusters through a single-cell approach. To this end, we used three technologies: single-cell RNA-seq for cell type identification, site-seq for cell type refinement, and TCR sequencing for functional annotation. To this end, we use information extracted from four diabetic mice and 4 healthy mice. First, we did all the single-cell RNA-seq pre-processing steps, and the cluster annotation was supported by SAC-seq information. Second, we performed compositional analysis and differential expression analysis to extract the first insight related to type 1 diabetes. And third, we use the TCR sequencing data to perform clonotype identification, and we use them to establish functional annotation based on database consulting. Here, we present the annotation of 27 identified clusters. However, today we will present the interpretation of 3 CD8 positive clusters and 4 CD4 positive clusters, focusing our attention into 2 annotated as regulatory T cells. Regarding the CD8 positive clusters, according to compositional analysis, effector CD8 were significant. Our interpretation is the change in composition is due to the activation of autoimmune-related T cells. Looking for supportive evidences, we analyze the gene expression and we observe a signature of T cell differentiation mainly into memory T cells. In addition, we observe a possible signal of residency given the downregulation of the gene THY1, a marker particularly abundant in peripheral T cells. Regarding TCR sequencing, the top 10 most abundant T cell receptors were identified in this cluster, supporting the hypothesis of high activation of effector cells in type 1 diabetes. Surprisingly, we were able to identify 4 receptors previously described in type 1 diabetes in humans. Here, we are presenting evidences about key receptors in type 1 diabetes interspecies. So far, we have presented results related to the expected behavior of CD8 positive effector cells. Now, when we analyze the composition of CD4 positive clusters, the T central memory and 1 T regulatory cluster rise as significant. For the next step, we focus our attention in the T regulatory cluster to try to identify key misfunctionalities in type 1 diabetes regulation. Analyzing the gene expression signature for the previously significant Treg, we were only able to detect the activation of this cluster mediated by the immunological signals. In addition, this process could be controlled by cell cycle stimulation. However, when we observe the second regulatory T cell cluster, we observe the differential expression of key autoimmune control-related genes. Here, we want to highlight the downregulation of the gene LAG3, which lack of expression has been associated with the acceleration of type 1 diabetes with 100% of incidence. Even this cluster shows specific signature for a regulatory subtype which specific function is the regulation of tissue autoimmunity. Furthermore, we identify 18 public clonotypes exclusively detected in the diabetic samples. They could be key factors related to autoimmunity in type 1 diabetes. Even more, 4 of them were previously detected in human type 1 diabetes. It means they could be potential key factors in type 1 diabetes in general, at interspecies level. We want to highlight 2 T cell receptors with additional information about epitopes. They will be focus of attention in our further analysis. We were interested in testing a particular hypothesis about the plasticity of Tregs in autoimmune diseases. Basically, the explanation is Tregs are not stable and they are prone to become into effector stage. To this end, we used the clonotypes as barcode to trace the change between Tregs and effectors. In the first Treg cluster, we identify one clonotype showing this change between Tregs and effector. However, when we observe the Treg cluster with clear tissue immunity regulation, we observe an active change between Treg and effector. This could be an additional factor related to type 1 diabetes beyond cell misfunctionalities. Finally, I want to share with you some take-home messages. First, we identify a clear signal of T regulatory related to autoimmunity control. However, we identify as well a gene expression supporting the functionality loss of this cluster. Second, we identify the lack of stability in the Treg cell fate. This is the loss of regulatory capacity in type 1 diabetes instead of a capacity for preventing it. Finally, the conclusion is beyond the misfunction due to gene expression impairment, there is a cellular behavior in favor of type 1 diabetes development. I want to say thank you for my partners at Venice Lab. Thank you to my colleagues at Computational Health Department, for our collaborators at Institute of Diabetes Research, and thanks to you for attending.